My name is Katharina Alamany. I'll be doing my presentation on glycolysis and gluconeogenesis. Glycolysis is the process in which glucose is broken down to produce energy. It produces two molecules of pyruvate, ATP, NADH, and water. The process takes place in the cytoplasm of a cell and does not require oxygen. It occurs in both aerobic and anaerobic organisms. Glycolysis is the primary step of cellular respiration, which occurs in all organisms. Glycolysis in following by the Krebs cycle during the aerobic respiration. In the absence of oxygen, the cells make small amounts of ATP as glycolysis is followed by fermentation. This metabolic pathway was discovered by three German biochemists in the early 19th century and is known as the EMP pathway. The glycolysis pathway occurs in the following stages. Stage 1. A phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase. In this, a phosphate group is transferred from ATP to glucose, forming glucose 6-phosphate. Stage 2. Glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme phosphoglucomutase. Stage 3. The other ATP molecule transfers a phosphate group to fructose 6-phosphate and converts it into fructose 1,6-bisophosphate by the action of the enzyme phosphofructokinase. Stage 4. The enzyme aldolase converts fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. Step 5. Triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. Step 6. This step undergoes two reactions. The enzyme glyceraldehyde 3-phosphate dehydrogenase transfers one hydrogen molecule to glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH plus H ion. In the second part, glyceraldehyde 3-phosphate dehydrogenase adds a phosphate to the oxidized glyceraldehyde phosphate to form 1,3-biphosphogylacerate. After that, there is step 7. Phosphate is transferred from 1,3-biphosphogylacerate to ADP to form ATP with the help of phosphoglycerokinase. Thus, two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. In step 8, we see the phosphate of both the phosphoglycerate molecule is relocated from the third to the second carbon to yield two molecules of 2-phosphoglycerate by the enzyme phosphoglycerate mutase. Step 9. The enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenolpyruvate. Step 10. A phosphate from phosphoenolpyruvate is transformed to ADP to form pyruvate and ATP by the end of the pyruvate kinase. Two molecules of pyruvate and ATP are obtained at the end of this product. Gluconeogenesis refers to synthesis of new glucose from non-carbohydrate precursors. Provides glucose when dietary intake is insufficient or absent. It also is essential in the regulation of acid-base balance, amino acid metabolism, and synthesis of carbohydrate-derived structural components. Gluconeogenesis occurs in liver and kidneys. The precursor of gluconeogenesis is lactate, glycerol, amino acids, and with propionate making a minor contribution. The gluconeogenesis pathway consumes ATP, which is derived primarily from the oxidation of fatty acids. The pathway uses several enzymes of glycolysis, with the exception of the enzymes of the irreversible step named pyruvate kinase, 6-phosphofructokinase, and hexokinase. The irreversible reactions of glycolysis are bypassed by the four alternate unique reactions of gluconeogenesis. The four unique reactions are pyruvate carboxylase, located in the mitochondrial matrix, phosphoenolpyruvate (PEP) carboxygenase, located in mitochondrial matrix, and cytosol, fructose 1,6 biphosphate located in the cytosol, and glucose 6-phosphatase located in the endoplasmic reticulum. Aldolase is a glycolic enzyme that catalyzes the conversion of fructose 1,6-diphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate via the glycolysis metabolic pathway. Within the cell, aldolase is localized in both the cytoplasm and the nucleus. 3 aldolase isoenzymes have been identified, A, B, and C, of which the muscle type aldolase A is found in the actin-containing filaments of the cytoskeleton. The enzyme regulates cell contractions through its reversible binding of these filaments. Aldolase is likely ubiquitous to all cells. The larger concentration present in muscles, liver, and brain cells. Increases the aldolase levels have been detected in myotonic muscle disease. Serum levels of aldolase also increase following acute coronary syndromes. Increases in aldolase A isoenzyme are primarily associated with these type of muscle injury. At present, aldolase is not widely used as a biomarker of skeletal muscle injury. Aldolase splits fructose 1 phosphate to dihydroxyacetone phosphate, and glyceraldehyde substrates for gluconeogenesis via fructose 1,6-diphosphate. Among the class 1 enzymes found in mammals, there are the three specific isozymes of aldolase that we were talking about. They have a similar molecular mass and catalytic mechanism. Aldolase A, expressed primarily in muscles and red blood cells. Aldolase B, expressed primarily in liver, kidney, and small intestine. And aldolase C, expressed primarily in brain, smooth muscle, and neuronal tissues. The aldolase isozymes are similar in sequence, with 66% identity between A and B, 68% identical of B and C, and 78% between A and C. All the isozymes have strictly conserved residues in the active sites. They are immunochemically distinct and have different catalytic properties, such as the ratio of specific constants. For the two hexose substrates of aldolase C is between those of A and B. Aldolases A and B have well-defined roles in glycolysis and gluconeogenesis, but the physiological role of aldolase C is still not widely known. Aldolase C is found in actively proliferating and cancer cells, and has been identified as zeverin in fetal rodent brain. Its gene expression is enhanced by nerve growth factor inducing B factor, which is transcription factor of the thyroid, steroid, retinoid, nuclear receptor gene family. This expression in rat brain embryo parallels that of rat aldolase C. Together, these results suggest a neurodevelopmental role of aldolase C, but biochemical data are not widely available to identify its physiological role. Thus, its structure may offer insight into the role of cell growth, vertebrate development, and other metabolic processes. The structures of rabbit muscle aldolase A, human muscle A, and human liver aldolase B demonstrate that these vertebrate enzymes are structurally similar. All possess a hemotetrameric alpha-beta barrel structure with the active site of each monomer located at the center of the barrel. It should be noted that the last 20 amino acids of the C-terminus is visualized in only a few of the published aldolase structures, that is observed in different conformations even with an isozyme. One of these conformations shows that C-terminus forms an arm-like structure attached by the highly flexible hinge that wraps around the surface, sometimes contacting the active site, while the ablation of the C-terminus produces an essentially inactive enzyme. The coupling mechanism between the catalytic cycle and the conformation change of the C-terminus remains unclear. Aldolase not only plays a key role in glycolysis, but also binds to macromolecules unrelated to glycolysis such as F-actin, vacuolar H plus ATPase, GLUT4, cell surface adhesins, and thrombospondin-related anonymous proteins, as well as others. Thus, aldolase may be used for other functions in the cell, a concept that has been described as moonlighting. These interactions may be specific to each isozyme. For example, studies have shown that aldolase associates with target proteins in the tissue-specific manner. Aldolase B binds to the liver cytoskeleton with higher affinity than to brain or muscle cytoskeleton. Aldolase C binds less tightly to the cytoskeleton compared to the other two, and this has been attributed to a more acidic environment. HNF1 and DBP are strong activators of aldolase B, which means the gene expression will be increased. HNF3 competitively inhibits HNF1 and non-competitively inhibits DPB. Either way, HNF3 decreases the expression of aldolase B. Lastly, CEBP are found weakly activated aldolase B in HepG2 cells and strongly activate aldolase B in hepatocytes in primary cultures, which are freshly harvested and exposed to growth media of the liver cell.